Hi everybody and welcome to this presentation by the MAVE Education Project. I'm going to share with you our presentation from the Functional Evidence Evaluation Workshop held on the 19th of June 2024. So first of all, I'd like to thank MAVE for supporting this MAVE Education Project, along with the collaborators that have really helped to develop this workshop and the project outcomes, the Atlas of Variant Effects, the Australian Functional Genomics Network, and ShareInt, the Australian National Genetic Variation Sharing Platform. So what is functional data? Kanavi in 2019 defined functional assays as systematic experiments, either in vitro or in vivo, that are used to elucidate the function of a protein in a cellular pathway or biological process. So functional assays can be quite wide and quite varied. You can see functional assays that use patient material, such as this alpha-gal assay. Some are looking for specific genetic variations. They can be studied in model systems from yeast and cell assays all the way through to mouse or animal models. They can also be low throughput to very high throughput. So on the left, you can see a single or three variant assay done in a cell line, which is a low throughput assay, through to on the right where you can see what is a saturation genome scanning assay, known as a MAVE or a multiplex assay of variant effect, which has very high throughput. So functional data is specifically different from functional evidence. We want to make a point here that with respect to variant curation, functional data refers to the experimentally derived data that indicates if a variant is impacting biological function, versus functional evidence, which is functional data that's been translated into weighted evidence to support variant classification. So we need to support the translation of functional data into functional evidence. So the first step is actually to find that functional evidence. And you can see here a list of sources of functional assays or functional data that you can evaluate to use in variant curation. And we also want to bring your attention to the recommendations for translating functional data into evidence. A paper published by Nitsch 2019 from the ClinGen Sequence Variant Interpretation Working Group. And this manuscript describes some recommendations for how you might consider a functional assay to determine what level of evidence you might apply in variant classification. So breaking this flowchart that was presented in Nitsch down to some very simplified questions, first of all, you want to consider if the assay measures the correct function. Does that assay measure the disease mechanism that you're hoping to investigate? Then you want to consider if the assay performance is sufficiently predictive for clinical application. And finally, you want to determine the evidence strength that can be applied for PS3 or BS3 for that assay. So if you're trying to consider if the assay measures the correct function, there's some things that you need to think about. Specifically, what is the disease mechanism? So there's a few different disease mechanisms that you might want to think about. Is the variant loss of function? These are variants that break the structure and function of the encoded protein. And some examples of this include BRCA variants and a number of Mendelian pathogenic variants. Or you could have a gain of function variant. And these variants are variants that induce an increase in activity compared to the normal gene product or protein. So often this is by loss of an inhibitory activity. So this includes some classical oncogenic mutations like the KRAS G12V. There's also a dominant negative mechanism, and this is where a variant might interfere with the function of also the normal wild type copy. And this includes some TP53 missense variants, for example. So you want to consider if the assay is measuring a function that is relevant for this particular disease mechanism associated with your variant. You also want to consider if the assay performance is sufficiently predictive for clinical application. And otherwise, you also want to think about the quality of that assay. Is it a broadly accepted assay type? What were the replicates used, and did they have both experimental and biological replicates? Also, considering those replicates, what's the variation that you can see in the assay? Does it support that it's a quality assay? What basic controls were used, wild type or null? And can you identify the number of known variant controls, benign and pathogenic? And are there enough of those so that you can evaluate the assay for predictive performance? Also, are there performance metrics provided? Can you benchmark that assay to determine if it's performing at a level sufficient for the known assay performance? Also, when you consider that assay, you want to think about what evidence strength can be applied. And here you can see a kind of simplified version of the recommendations in the SVI recommendations manuscript that we shared before. So have a look at the number of control variants. And by this, we're meaning the assumed benign and pathogenic reference sets that are being used to determine the clinical predictive power of that assay. Do you have sufficient variance to calculate an odds path? So if you don't, then if you have less than 10 control variants, then the SVI recommendations would suggest that you should apply a maximum of PS3 or supporting evidence. If you have more than 11, then you may be able to apply moderate or at least higher than supporting evidence. If you can calculate an odds path, if you have sufficient benign and pathogenic reference variance, then you should calculate the odds path and, or the likelihood ratio and determine what actual evidence strength that you can apply for classification of that variant. So this could be anywhere from BS3 to BS strong, depending on the likelihood ratio that you calculate. So high throughput assays are something we're going to talk about today because these have been identified by our workshop participants in the past as an area where they'd like to have more information and be able to evaluate with a little bit more confidence. So we're talking about high throughput assays, but this includes a variety of different types of assays. There's massively parallel reporter assays, deep mutational scanning, and MAVEs, which are multiplex assays of variant effect. MAVEs perform saturation mutagenesis. In other words, they look at the impact of every single variant within a particular genomic region. But the benefits of high throughput assays are similar across all of these assays because you can look at that across the population of variants you're investigating to look at the predictive power of that assay. So there's a lot of value in evaluating and applying functional evidence based on the evidence produced in high throughput assays. So it is also a rapidly building field. There are more and more high throughput functional assays. You can see this by the increasing number of assay score sets that are in MAVE-DB, the database of MAVE assays. MAVE-DB is also building a number of different use cases. So they have a UCSC track, and soon there'll be a MAVE-DB clinical variants page. It's also the Atlas of Variant Effects, which is an international community. They're highly collaborative, and they're working on updating guidelines and means to share and support generating and sharing and using variant-specific evidence strength, especially in high-throughput assays. So along with these growing communities, there's developing recommendations and guidelines for high-throughput assays. Highly recommend you look at the Gelman 2019 paper we've listed here, which has recommendations on how to consider quality and how to evaluate MAVE assays. Also, there's a MAVE minimum information and reporting guidelines, which are important for how to understand language around MAVEs, but also simply on how to build these quality and shareable data sets. There's also a couple of other papers there which do help you understand some of the considerations around MAVE assays. And if you're interested in evaluating and applying MAVE assays, I suggest you have a look at these. I want to specifically point out some of the recommendations within Gelman 2019. So while this paper is research and assay design and development focused, it does cover the assay design and reporting as well. So there's 12 recommendations, but I want to point out these specifically pertinent recommendations. So they recommend in this manuscript to use standard terms for control variants, benign and pathogenic. They also suggest that you should report the predictive value for each gene disease pair for your assay. And also they do point out that not to stack evidence. So we'll get back to this when we talk about combining assay evidence. Now, it's important to note that in this paper, they recommend variant-specific evidence strength calculation, and this is not really standard field practice yet. And we're going to show you how to calculate category-specific evidence strength through an assay and how you apply that for a specific variant. So High throughput assays, how do they work? So usually a number of different assays are introduced, and the output is a score for each of those specific variants that you've assayed. So on the right, you can see the distribution of the functional scores in this Findlay assay. So you can also see in this assay that the scores have been categorized into three separate categories: loss of function, intermediate, or functional. So with a high throughput assay, you can calculate how predictive the assay is, or how predictive each of those assay category is, using the reference set. So this is according to a kind of a Bayesian framework as published by Tijian in 2018. So in this paper, they calculated the odds of pathogenicity, which is the combined measure of probability of the test result being correctly associated with that pathogenic category. And we can compare the estimated likelihood ratio for this specific assay to the odds of pathogenicity, determine how strong or how predictive that assay is. So I'm going to go into this a little bit. We can use this high throughput assay. We've got the example of Findlay here to calculate a category associated with functional impact, and then we can calculate the likelihood ratio for each of those functional categories. So in this case, we can have a look at the reference set, 169 pathogenic and 22 benign. And we can calculate how many of those variants have been correctly predicted. So pathogenic, for example, in the loss of function category. So 162 of the 169 pathogenic variants have been correctly predicted as loss of function. 20 of the 22 benign variants have been correctly predicted as functional. So we can calculate the likelihood ratio for each of these categories. I'm going to show you the calculation for the loss of function category. So you can see here that 162 of the 169 over the one variant that has been incorrectly predicted as benign to calculate a likelihood ratio of 21.9. So we can then compare this to the odds of pathogenicity thresholds that indicate evidence strength as calculated by the Bayesian framework in 2018. So a likelihood ratio of 21.9 is above the odds pathogenicity threshold of 18.7. In other words, a likelihood ratio of 21.9 correlates to an evidence strength of PS3, or strong evidence. So if we're looking at a specific variant in this assay, for example, this BRCA1 variant here, P1749R, it scores a functional score of -3.3. This is well within the loss of function category. And this category we've just calculated has a likelihood ratio of 21.9. This means that this category has a PS3 strong. So a variant that scores well within the loss of function category can have an evidence strength of PS3 or strong applied in variant classification. So that's how you do one assay. What considerations have we got when we're thinking about this in a broader sense? So what do you do if you have multiple assays? In Benich 2019 and Gelman, they both mention stacking evidence and essentially say that if the results are consistent, apply PS3 and BS3 at a level of strength appropriate for the most well-validated assay. In other words, if the evidence is in the same direction, supporting each other, don't sum evidence for the same class. The highest evidence wins. Cap evidence in the same direction from multiple assays to max strong. This will hopefully prevent you from overweighting the functional evidence. Now, what about if the assays are not in the same direction, if the results are conflicting? At this point, there's no clear advice, but they are being developed. There's research projects looking to develop advice in this space. So the recommendations at this point say that if they're conflicting, apply evidence according to the best assay evidence, unless it's the same level of validation or quality, in which case maybe don't use the functional code. There's not a good consensus on how to combine the different assay classes, for example. So you just have to use your curator and expert opinion to be able to decide what's the appropriate action in this case. In some possibilities, if the evidence is conflicting between assays, you should Definitely review the assay class design and the mechanism of impact so that you can identify if that assay really is the appropriate assay for that mechanism of disease. For example, a splicogenic variant will show impact in an assay that measures RNA, but it won't show in an assay that measures protein impacts only. So does your assay pick up all of the potential impacts that you might need to be measuring? Some assays don't pick up all protein-related impacts. For example, some will detect binding impacts versus subcellular localization. So you could also consider just not using functional data in these conflicting cases, or to some as negative and positive points. If you have multiple assays that you can calibrate, you could consider combining the assays and then recalibrating to determine which category has, which summed category has which predictive power and evidence strength. So I'll just quickly go through some case examples of variants with some considerations that you can keep in mind when you're evaluating functional assays. There are many well-known, well-validated low-throughput assays. Don't be put off by an assay that's only looked at one or two variants. You can see this HNF1 variant, which has been assayed for transcriptional activation via a reporter gene, or this one on the right, which is looking in a cell line for just for a handful of PAH enzyme variants. However, in some cases, low throughput assays do require some careful considerations. Are they reliable assays? Do they have controls? Do the controls have a large range? Also, are the pathogenic variants that they're investigating rare or common? Some assays also investigate the residue and not the variant. For example, this assay example you can see here, where it's looking at phosphorylation of that residue, but doesn't investigate the impact of a variant at that residue on SMAD3. You can also predict functional evidence, not just for the pathogenic category, but also for benign categories if you have sufficient predictive power in your assay. So this assay I showed you before, where we calculated the likelihood ratio for the loss of function category. This variant in BRCA1, you can see has a score of -1.8, is in the loss of function category, and therefore you can apply PS3 evidence. However, for using the same assay, this variant, which scores 0.455, minus 0.58, scores within the functional category, which is calculated to have a likelihood ratio of 0.01. And this is sufficient for benign evidence, or BS3. So you can apply benign evidence in the case of an assay that you've evaluated to be sufficiently predictive to do so. You do specifically want to be careful that you've captured the variant's mechanism, though, if you're inferring that this variant is still functional and therefore eligible for benign evidence. Now, in some cases, in an assay that you can calculate the predictive power, there are variants at which there's not sufficient evidence to apply either PS3 or BS3. For example, this variant here, which scores in the intermediate category for the assay that we've been looking at before. And this variant scoring in the intermediate category doesn't have sufficient predictive power to apply evidence either way, pathogenic or benign. In this case, you just don't apply evidence. In some assays, there's uncertainty in the clinical reference set. So if there's insufficient pathogenic or benign variants to calculate the likelihood ratio, too few controls, in other words, then you really want to consider whether or not you can apply evidence in this case. Or if you want to apply evidence at a lower evidence strength. So finally, you also want to consider if you're detecting the right mechanism and which specific mechanism you're detecting. Splicing variants in particular, you want to consider what the assay is detecting. The first assay you can see here is an assay that validates the splicing impact. So you can see the alternative splice form in this case after introduction of the variant, versus an assay that can detect loss of function due to a splice impacting variant. So this is an assay that uses a method of assay that will have an impact from a splice impacting variant, and therefore you will detect a loss of function due to a splicing variant. So you want to consider whether or not the assay you're looking at is validating the splicing or validating the splicing impact. So finally, I just want to remind you that there are recommendations for translating functional data into functional evidence. And essentially, this breaks down into, first of all, deciding if the assay is measuring the appropriate disease mechanism. Then, is that assay high enough quality and sufficiently predictive for clinical application? And can you calculate the evidence strength for that assay? What evidence strength then can be applied for BS3 or PS3 or functional evidence? So to summarise, functional assays must be evaluated and the evidence strength determined before you apply them as functional evidence. The evidence evaluation is complex, but it's a rapidly developing field. So look for some of the coming publications. There are recommendations available, and specifically the ClinGen SVI group, functional evidence recommendations, and the MAVE recommendations are an excellent source to start building your confidence in functional evidence evaluation. There is general support to improve tools and research methods too for functional evidence evaluation. And so in the MAVE Education Project, we're going to keep working on what tools and methods we can support you with in your functional evidence application. So coming soon, we have an updated likelihood ratio calculator. So based on the comments that you've provided within the workshop, we'll update the likelihood ratio calculator and share it with the participants. And so far within the MAVE Education Project, you have supported the expert-reviewed functional assays, tools to support evidence evaluation, and increased accessible training and practice opportunities for functional evidence evaluation. And just to bring it to your attention that both MAVE-DB and the Atlas of Variant Effects are working to build support in the areas, and there will be a MAVE-DB clinical page coming soon, which should help you determine the functional evidence that's evaluation for your particular assay and how it might apply for a specific variant. So the future of functional evidence is bright. The international field is working across methods for analysing high throughput assays and how to combine results and support clinical evaluation. There's large projects delivering many high throughput assays, so get ready. It's a rapidly growing field. And I just want to finish with a statement that from a publication by Feller and Riemann that claimed that most VUS encoding regions will be resolved by 2030. And I want to leave you with this thought and with the prospect that these high throughput functional assays might contribute to resolving VUS in the future. So I just want to thank the project leads, Professor Spurtle and Dr. Rubin, and also the AVE team for really supporting this work. I also want to thank a lot of the workshop facilitators who've been involved in building the resources that we've shared with you today. And finally, I want to thank the MAVE Education Project Working Group. All of these members have been integral in putting these resources and the continuing resources that we're building within the MAVE Education Project. And thank you for listening.